Okay, so we're going to use this figure, which is in your textbook, to summarize basically step by step how do we get from a signal from your brain all the way to a muscle contraction. Then we're also going to see how the muscle will then relax. But we're going to piece this whole thing apart a little bit. There's a lot going on here, but we're going to kind of break it down into 3 steps. We're going to talk about how a neuron stimulates the muscle. Then we're going to see how that impacts the sarcomeres. And then how does that muscle relax? So we're going to contract and then relax. All right. So we're actually going to start with this part here, which is called the neuromuscular junction. So a neuromuscular junction is the junction between a neuron and the muscle cell. So that's right here. All right, so step by step. So first of all, we really start at the brain, right? All skeletal muscle, unless it's a reflex, gets stimulated because there's a signal that comes from your brain. That's why these are voluntary muscle contractions. So a signal starts in your brain, and that signal is called an action potential. So we'll learn a little more about what an action potential is when we get to nervous system. But, another name for an action potential is an impulse or an electrical signal. And these electrical signals or action potentials are due to ions, all right? Ions going in and out of the cell through the plasma membrane, okay? But for now, we can just think of it as a nerve impulse. So there's a nerve impulse or action potential that travels down the neuron. All right, so this is what's called the axon of a neuron. What's the neuron's specific name? It's a somatic motor neuron. Somatic motor neurons stimulate skeletal muscle. All right, somatic means body, and motor means, you're kind of moving it. So somatic motor neurons control skeletal muscle that moves the body. All right, so that's what the neurons are called. Now, when the signal gets to the end of the axon, this end of the axon is called the synaptic end bulb, because it's kind of like a bulb here. This figure calls it a presynaptic terminal. But anyway, it's the end of the axon. All right, so this AP represents the action potential. So the signal comes down, and that signal is going to open up what's called an ion channel. And this ion channel is actually a voltage-gated calcium channel. So what does that mean? Well, it means the only way it opens is if there's a voltage. And the voltage is the action potential. So the action potential opens the channel. Calcium enters. All right, so this is the second role for calcium. We're going to learn 2 roles for calcium. 1, we talked about it binding to troponin. 2, it's required for exocytosis. All right, inside this end bulb are what's known as synaptic vesicles. These are synaptic vesicles. They're basically containers of neurotransmitter. So these little yellow and orange spheres in here are the neurotransmitter that's going to stimulate skeletal muscle. And that neurotransmitter has to get exocytosed. Remember, we learned about that back in chapter 3 when we learned about ways to move things out of a cell. This process is exocytosis of the synaptic vesicles. What that does is it releases ACH. Well, ACH is short for acetylcholine. Acetylcholine is the neurotransmitter that stimulates skeletal muscle contraction. All right, so it gets released from the neuron. It has to attach to a receptor on the muscle cell. All right, so this is the acetylcholine receptor. All right, so it has to travel across this little gap. This little gap is called the synaptic cleft. Cleft is a gap. All right, so it crosses that little gap or cleft, attaches to the receptor, and the receptor is a sodium channel. Now it's known as a ligand gated because acetylcholine is a ligand. A ligand is basically a chemical. So now instead of opening due to a voltage like this channel, it's opening because of a ligand or chemical called acetylcholine. Once acetylcholine attaches, the channel opens and it lets sodium enter the muscle cell. Now, sodium is a positive ion. It causes an action potential in the muscle. So notice now we have this arrow here, this AP. So there's electrical signals that travel down the neuron. There's also an electrical signal that travels along the sarcolemma of the muscle cell. All right? So acetylcholine binds to ligand-gated sodium channels. Those are the acetylcholine receptors. They're the same thing. All right? They're on the motor end plate. That's this part of the muscle cell where you have the receptors. Lets in the sodium. The sodium is going to cause a muscle action potential. So that's shown here with this arrow. All right? So now we've got a signal traveling along that membrane. Okay? Now what's going to happen next? Okay, what happens next is here it is traveling along the membrane. All right, now what is an action potential? Again, we'll talk more about it in neurons, but it's basically the entry of sodium and the exiting of potassium. All right, so that's what an action potential is. Sodium enters, potassium exits. And that keeps happening along the membrane. Now notice what we're doing. Now we're going to go down a T-tubule. All right, so this is that T-tubule part. So why did we go down a T-tubule? Well, we wanted to get this signal close enough to the sarcoplasmic reticulum. So this signal that travels down here results in the release of calcium. All right, so calcium gets released from the sarcoplasmic reticulum through little channels. All right, they don't really show the channels here, but calcium comes out through little voltage-gated calcium channels. And then the calcium is going to go find troponin. All right, remember calcium binds to troponin. When calcium binds to troponin, that's going to move tropomyosin so you can form cross bridges. All right, and once you form the cross bridges, you can contract the sarcomere. All right, so calcium binds troponin. Binding of calcium to troponin causes tropomyosin to move. Remember, tropomyosin is the one that blocks. We got to move tropomyosin. That's going to expose the binding sites. So now you can bind myosin to the actin, form that cross bridge, and as long as there's energy, it's going to cause that ratcheting, and that sarcomere is going to shorten or contract. And if you get all those sarcomeres to shorten inside of a muscle cell, then that entire muscle cell is going to contract. And if you contract all the muscle cells inside of a muscle, well, then that muscle will contract. All right? So that's kind of putting it all together. All right? All the way up until we cause muscle contraction. Now the question is, how do you relax? All right? So that's what we're going to talk about here. So this is inhibition or relaxation of the muscle. So first and foremost, the neuron stops sending the signal. That's the first thing you do. The signal stops coming from your brain. All right? Once the signal stops coming from your brain, acetylcholine is going to detach. So that's shown right here. So we have detachment of acetylcholine. Then acetylcholine gets broken down. So there's an enzyme in here. So here's this little enzyme shown here in green called acetylcholinesterase. So acetylcholinesterase breaks down acetylcholine because you got to get rid of it. If you leave the acetylcholine floating around in here, you're going to keep stimulating the muscle. So you got to break it down. You got to get rid of it. All right? And you actually recycle the choline. So acetylcholine is part acetyl and part choline. All right, so you actually shown here, the yellow part of it is the choline, And when it gets broken there, the choline gets recycled so you can reform acetylcholine. And then the acetic acid, notice that kind of just diffuses away. So you reabsorb in the choline so you can reform acetylcholine very quickly. So now you can make more acetylcholine, but it's packaged, it's repackaged in the synaptic vesicles. So now you're no longer stimulating the muscle. All right. Then you got to remove that calcium. So when the muscle cell is no longer being stimulated, calcium detaches from troponin. The cross bridges are blocked by tropomyosin, and the muscle will relax. All right. So the last 3 slides, we went from stimulating the muscle cell, causing it to contract, and now we just caused it to relax again. And that's what muscles do. They contract and relax. It just so happens there's a lot of little molecules that play roles during that process. All right, so I'll remind you once again, it's really hard to learn this by staring at pictures. You got to see it working. And the best way to see it happen are watch the animations. You might want to have this up in front of you. And while you watch the animations, you can kind of follow these steps through the process. The next video, we're going to talk about some other things like rigor mortis. Once you know how muscle contracts, you can understand why when animals die, they become stiff. So we'll talk about that. We're going to talk about some other things that might affect the neuromuscular junction and therefore affect whether muscle's going to contract or not. And then we're going to move into a few other topics. All right, so we'll stop this video and start another one here in a minute.